So this is our plate replicating device. This is pretty standard. It's just a circular piece of hard plastic with a ring on it. And we're going to put something onto the top of this which is sterile and allows the cells to be contacted to that and then transfer to the next plates. And we're actually going to use something that is effectively sterile. So these days there are factories that are automated and they manufacture things like paper towels pretty quickly in large numbers. And the chance that something, a little bacterial cell, gets in there in that part of the process is actually pretty low. So we're going to treat this as effectively sterile. If you wanted to be really sterile, you could autoclave something like this. If you worked in a technical yeast lab, you would certainly autoclave your transfer sheet. And they're usually made out of velvet, so maybe I'll show you that sometime. For us, we're going to take the inside of this. So obviously the outside, I've been touching it with my hands. It's been exposed to the air. But the inside, we're going to assume has been pretty well concealed in there. And we're going to try not to touch it. So I'm going to touch just the edges here. Roll this. Put it onto the device. And kind of carefully find my circle there. And just put it down on the collar. So this holds everybody in place. And it's just the shape of the Petri dishes that we're going to copy. So a couple things. We're going to do things sort of upside down. So I'm going to leave this block of plastic here, and I'm going to add the plates to it. I'm going to make a mark on here to orient myself. So when you're working with circular Petri dishes, you might not know which way is left, right, up, down, and so on. And I'm going to make a corresponding mark on the plates. It really doesn't matter where, but you'll just have a point of comparison for these different plates. So my source and my target plates are going to have this mark on them. The formamide plates also have a red mark on there that tells us that they're formamide. And I just see that I've got red marks on both sides. So sometimes I would use the red mark to orient ourselves. The fact that there are two red marks makes that a little less effective. All right, so we're going to line up our black marks on the starting plate to transfer cells from, and set that down so it's on there loosely right now. And I'm just going to do a little finger tap dance on there and get the paper towel to contact all parts of that auger there. Take this off, and you'll see that there's kind of a ghost of where the colonies used to be. There still are plenty of cells on here. And the thing about growing up cells is you just need the tiniest amount. So you don't have to see them get transferred to know that they're there, or I guess to believe that they're there. I'm going to do the formamide one first, just to increase my chances of finding any. If anybody didn't make the transfer onto formamide, then I'll see that as I plate them onto the next plate. So tap dance, kind of the same thing. You want to, as you're doing lab techniques, if they involve human operation, you want to make your human operations as reproducible as possible. So kind of get your technique down. Put them on there. You can even see there's enough cells in some places where you can see that I've transferred the cells. So those are going to grow up on the formamide plates. And then finally our YPD control to make sure that cells can actually get transferred. So tap, 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 tap. And we'll see anybody. Look at that. It's so effective. I can still see kind of the same ghost pattern on that plate. So let me hold these up next to each other. We're actually not relying on anything that we're able to see, but just the fact that you can see them, I think, really increases our confidence that we transferred. So look at the same pattern on the places where I'm pointing there. We're going to grow those up in the incubator and see if any of these colonies have trouble growing. That would be evidence that they might be formamide sensitive. So I'll do this again with our other 4 replicates of that same dilution, see if we get any interesting mutants.